Welcome to our YouTube channel. Today we're delving into an essential molecular biology technique, high-fidelity PCR. This technique plays a crucial role in medical research and diagnostics. In this educational video, we'll provide an in-depth explanation of the principles and applications of high-fidelity PCR, helping you understand why it's so important and how to use it in the laboratory. Whether you're a medical professional or a science enthusiast, you'll benefit from today's content. Let's get started and explore this exciting technology together. Fidelity of a DNA polymerase. In the application of cloning, site-directed mutation, sequencing template preparation, and gene variability research, it is very important to maintain the accuracy of DNA sequence in PCR amplification. The ability of DNA polymerases to replicate DNA templates accurately is called fidelity. In the process of DNA synthesis, some polymerases can read and copy the sequence more accurately and preferentially to combine the correct nucleotide, so that they can integrate the correct nucleotide faster than other polymerases. In addition, DNA polymerases may also have the correction activity or exonuclease activity with 3' to 5' primers. When an incorrect base is added, the polymerase will recognize the error, and the wrong nucleotide will be removed and replaced with the correct one. The synthesis of the new DNA strand can then resume. Error probability is often used as an indicator to compare the fidelity of different DNA polymerases. When the mismatch ratio of nucleotide incorporation is more than 1 out of 1,000, it is a low-fidelity DNA polymerase. When the mismatch rate is less than 1 out of 1 million, it is high-fidelity DNA polymerase. Fidelity is usually expressed as a value relative to Taq DNA polymerase. Based on this comparison method, the second-generation sequencing method can be used to determine the fidelity of different DNA polymerases. What is high-fidelity PCR? High-fidelity PCR is a modification of conventional PCR, which utilizes a DNA polymerase with a low error rate and results in a high degree of accuracy in the replication of DNA of interest. Many high-fidelity polymerases like platinum superphi, PFU, Vent DNA polymerase have been discovered. They have different processivity, speed, and levels of accuracy. The new generation of high-fidelity enzymes for PCR amplification of DNA has become commercially available. It has been shown that fusion high-fidelity DNA polymerase, when used for DNA synthesis, has dramatically lower error rates compared to Taq and PFU. Some research showed that Q5 is approximately 200 times more faithful at replicating DNA than Taq. But results from Sanger sequencing showed that this value may actually be an underestimate. KOD is also an enzyme used in PCR experiments. Compared with Taq, it has low mismatch rate and high accuracy. It is very suitable for long fragment cloning. At the same time, because it is a hot start enzyme, it does not need ice bath to configure the reaction system. Figure 2 is an analysis of OE-PCR performance of 3 polymerases, Taq, Fusion, and PFU, using gel electrophoresis. DNA fragment corresponding to a full-sized FELD4 gene 581BP was synthesized by one-step PCR. PFU polymerase gave clearly visible band about 581 BP, as was anticipated, but there was a number of smeary diffuse bands above and one extra 300 BP band. Fusion polymerase produced one pure band, corresponding to the PCR product of the correct size. Certainly, here are some classic high-fidelity enzymes widely used in PCR and DNA amplification, known for their high-accurate DNA synthesis capabilities. PFU DNA polymerase. PFU DNA polymerase is a high-fidelity enzyme with 3' to 5' exonuclease activity, enabling self-correction and reducing error rates during PCR. It is widely used in gene cloning and DNA sequencing. Fusion DNA polymerase. Fusion DNA polymerase is another highly refined enzyme with highly accurate DNA synthesis capabilities. It combines the high fidelity of PFU enzyme and the rapid amplification rate of Taq polymerase, making it suitable for various PCR applications. KOD DNA polymerase. Derived from Thermococcus codocarensis. KOD DNA polymerase exhibits exceptional heat stability and high fidelity. It performs well at high temperature conditions and is suitable for specific PCR applications. Taq DNA polymerase. Although Taq polymerase is not a high-fidelity enzyme, it is one of the most commonly used DNA polymerases in PCR. It has a relatively high amplification speed, but is prone to errors, so it is often combined with proofreading enzymes in some applications to enhance accuracy. How to determine the fidelity of polymerase. Many polymerase fidelity assays have been described over the years. The most famous method is called Kunkel. The Kunkel method uses portions of the LAC-Z alpha gene in M3 bacteriophage to correlate host bacterial colony color changes with errors in DNA synthesis. Another method is established by Wayne Barnes. This assay utilized PCR to copy the entire LAC-Z gene and portions of 2 drug-resistant genes with subsequent ligation, cloning, transformation, and blue-white colony color determination. In both assays, errors incorporated in the LAC-Z gene cause a disruption in beta-galactosidase activity leading to a white colony phenotype. In order to accurately compare the fidelity of different polymerases, we must use the same processing method to determine under the same reaction conditions. Different methods may lead to different results. Suppose you want to do a cloning experiment. A 1000 BP fragment was amplified by 25 cycles in the construction of PCR. If Taq DNA polymerase and specific PCR conditions are used, more than half of the clones are expected to contain a mismatch. Using proper DNA polymerase could make less mismatches and help to reduce the number of clones that need to be screened. To maintain the highest fidelity of polymerase, we should use high-fidelity reactive enzyme and optimized reaction buffer. Any additives should be avoided. Using fresh and high-quality DNTPs may also help to keep the high fidelity of polymerase. Other methods like limiting the number of cycles according to the minimum demand is also widely used.